Hello students, welcome to EPG Pathshala. I am Sabah Khan and we will be discussing today solubility and drug dissolution. In solubility and drug dissolution, what all we will be discussing about include BCS classification, dissolution testing objectives and applications, dissolution controlling parameters and factors affecting dissolution of drug. Then we will be talking about different types of dissolution apparatus, dissolution media and methods. Then we will also take a look on different approaches for enhancing drug dissolution. Now firstly, let's get familiar with the terms like solubility and drug dissolution. So solubility is the property of the substances to dissolve in given particular solvent. The solubility of a substance depends on the physical and chemical properties of the solute and solvent, temperature, pressure and the pH of the solution. So drug dissolution is defined as the process by which a known amount of drug substance goes into solution per unit time under standardized condition. The dissolution of a solid in a given solvent may be composed of two stages. First is an interfacial reaction that results in the liberation of solute molecule from the solid phase, which involves a phase change, so that molecule of solid become molecule of solute in the solvent in which the crystal is dissolving. Second step is the migration of the solute molecules through the boundary layer surrounding the crystal to the bulk of the solution and molecules away from the solid-liquid interface into the bulk of the liquid phase under the influence of diffusion. The Noyes-Whitney equation was developed to define the dissolution from a single spherical particle, which states that the rate of mass transfer of solute molecule or ion through a static diffusion layer, that is dm by dt, is directly proportional to the area available for molecule or ionic migration, the concentration difference across the boundary layer, and is inversely proportional to the thickness of the boundary layer, that is H. So this relationship can be seen in the equation. We can see that D is the diffusion coefficient or diffusivity of the drug, A is surface area of dissolving drug, K is water to oil partition coefficient of the drug, V is volume of dissolution medium, H is thickness of stagnant layer, and Cs minus Cb is concentration gradient of diffusion of the drug. A very important topic in solubility and drug dissolution is biopharmaceutical classification system. Drugs are classified into 4 classes depending upon their solubility across the gastrointestinal pH range and their permeability across the gastrointestinal mucosa. A drug is considered highly soluble when the highest strength is soluble in 250 ml or less of aqueous media over the pH range of 1 to 7.5. Similarly, a drug substance is considered highly permeable when the extent of intestinal absorption is determined to be 90%. So in BCS classification there are 4 classes: class 1, class 2, class 3 and class 4. In class 1, the drug is considered to be highly soluble and highly permeable. When the drug is having low solubility and high permeability, then it belongs to class 2 of BCS classification. When the drug is having high solubility and low permeability, then it belongs to class 3. And when the drug is having both low solubility and low permeability, then it falls under the class 4 of BCS classification system. Now, these class 1 drugs will dissolve rapidly when presented in immediate release dosage forms and are also rapidly transported across the gut wall. It is expected that such drugs will be rapidly absorbed and thus show good bioavailability. For drugs of class 2 of BCS, the dissolution rate is the rate limiting step in their oral absorption. Therefore, it should be possible to generate a strong correlation between in vitro dissolution and in vivo absorption. For class 3 drugs, these are those that dissolve rapidly but which are poorly permeable, and the class 4 drugs are those that are classed as poorly soluble and poorly permeable, as these drugs are liable to have poor oral bioavailability or the oral absorption may be slow as they cannot be given by oral route. Next is solubility as a rate limiting step to absorption of drugs. Solid drugs need to dissolve before they can be absorbed. A drug must possess aqueous solubility in excess of 10 mg per ml for therapeutic efficacy in physiological pH range of 1 to 8 at 37 degrees Celsius. Poor solubility may result into bioabsorption problem. If a drug solubility is in the range of 1 to 10 mg per ml, consideration should be given to the chemical modification of the drug into salt form so as to avoid potential absorption problems. Drug dissolution is a prerequisite to drug absorption and clinical response for almost all drugs given orally. For a drug to be absorbed into the bloodstream to reach its site of action, it should be present in a solution form in the GI tract, more specifically in the intestine. And for drugs having poor aqueous solubility, the lack of ability of drug to go into solution becomes the major limiting factor to its overall rate of absorption than its ability to permeate through intestinal mucosa. So for these kind of drugs that cross intestinal mucosa easily, the onset of drug level will be dictated by their time required for dosage form to release its content and its drug to dissolve, and its dissolution rate is so slow that dissolution takes longer transit time past its absorptive sites, which in turn results in lower bioavailability. Thus, for a drug which has a poor aqueous solubility, the rate at which it dissolves in the gastrointestinal fluid is often the slowest step, and the bioavailability of that drug is said to be dissolution rate limited. Now the next important topic is factors affecting dissolution rate. Now what are all the factors that affect dissolution rate of a drug? These are basically divided into two types. First are physiological factors and second are physicochemical factors. Physiological factors include surfactants in gastric juice and bile, pH, buffer capacity, bile, food components, permeability transit, viscosity of lumen contents, mobility patterns and flow rate, gastrointestinal secretions, co-administered fluid. And what comes under physicochemical factor? It is the particle size of drug or formulation, wettability, hydrophilicity, crystal structure, solubilization of drug, and its molecular size. Now the next topic is dissolution testing objectives and application. Dissolution testing is a standardized method for measuring the rate and extent of drug release from a dosage form. Dissolution is used as a tool to identify formulation factors that are influencing and may have crucial effect on the bioavailability of the drug. Dissolution test can be used to support the bioavailability of new drug product, the bioequivalence of an essentially similar product or variations. It is also used as a tool in quality control to demonstrate consistency in manufacture. Now the dissolution studies have several purposes. For example, quality assurance, to get information on the test batches used in bioavailability, bioequivalence studies, and pivotal clinical studies to support specification for quality control, to be used as a tool in quality control to demonstrate consistency in manufacture. These dissolution studies are also used to get information on the reference product used in bioavailability, bioequivalence studies, and pivotal clinical studies. They are also used to determine percentage drug release at preset sampling points and to meet legal requirement for compendial method. Next is bioequivalence inference. So these dissolution studies are also used to demonstrate similarity between reference products, to demonstrate similarity between different formulations of an active substance and the reference medicinal product, and to collect information on batch to batch consistency of the product to be used as basis for the selection of appropriate batches for in vivo study. Now the next very important topic is different types of dissolution apparatus. The table given in this slide describe the different types of dissolution apparatus and their suitable application. For example, first is basket apparatus, which is used for solid wheat powders. Then paddle apparatus, it is used for solid formulations. Then there is reciprocating cylinder, flow through cells, paddle over disc, rotating cylinder, reciprocating holder. These paddle over disc, rotating cylinder, they can be used for transdermal patches determination. So a very important type 1 apparatus, that is basket apparatus. In this apparatus consists of vessel made up of glass or other inert transparent material which is cylindrical in shape and having hemispherical bottom, and its capacity is 1 liter, 2 liter or 4 liter. A motor is there, a metallic drive shaft and a basket is there in this apparatus. The vessel is partially immersed in a suitable water bath to maintain temperature at 37 degrees Celsius plus minus 0.5 degrees Celsius. The shaft is positioned so that its axis is not more than 2 mm at any point from vertical axis of the vessel and rotates smoothly and is made up of stainless steel 316. A cylindrical basket made up of 22 mesh to hold dosage form is located centrally in the vessel in such a way that the distance between inside bottom of the vessel and basket is maintained at 25 mm plus minus 2 mm during the test. Next slide depicts the figure of basket apparatus. Next is USP type 2 or paddle apparatus. USP type 2 paddle apparatus uses the same assembly as that for apparatus 1 except that the rotating basket is replaced with a paddle. The dosage form is allowed to sink to the bottom of the vessel before the rotation of blade is started. A small loose piece of non-reactive material may be attached to the dosage form that would otherwise float. Next slide depicts the figure. Next is USP apparatus 3 or reciprocating cylinder. In this, apparatus is consist of a set of cylindrical flat bottomed glass vessel, a set of glass reciprocating cylinder, inert fittings made up of SS316 or other inert material, and screens made up of suitable non-absorbing and non-reactive materials which are designed to fit the tops and bottoms of the reciprocating cylinder. Next is USP apparatus 4, which is also called flow-through cell. This apparatus consists of reservoir and pump for the dissolution medium through the cell that holds the test sample and a water bath that maintains the dissolution medium at 37 degrees Celsius. Pump has flow rates of 4, 8 and 16 milliliter per minute. In USP apparatus 5, type 5 apparatus, which is also called as paddle over disc. In this, the assembly is used for testing the release of drug from transdermal product. It consists of sample holder or disc assembly holding product, which is then placed in a dissolution flask filled with specified medium maintained at 32 degrees Celsius. The paddle is placed directly over the disc assembly. Sampling is done at specified times. Next is USP type 6 or cylindrical method. In this, the assembly is exactly similar as apparatus 1 except that the basket and shaft is replaced with stainless steel cylinder, stirring element, and to maintain the temperature at 32 degrees Celsius during the test. The shaft and cylinder component of the stirring element are fabricated of stainless steel. Next, USP type 7 or reciprocating holder. This assembly consists of a set of a volumetrically calibrated or tiered solutions containers made up of glass or other suitable inert material, a motor and drive assembly to reciprocate the system and a set of suitable sample holders. Next, we will be talking about dissolution media and methods. Now, the selection of dissolution medium should be based on physical and chemical data for drug substance and dosage form, such as solubility and solution state stability of the drug. Evaluation of the influence of buffers, pH value and surfactants on the solubility and stability of drug should be carried out for the selection of the composition of the medium. Now, for immediate release products with water soluble drug, water or phosphate buffer pH 6.8 can be used as appropriate dissolution medium, and dissolution is carried out using either type 1 or type 2 apparatus and conditions set at 37 degrees Celsius 50 to 100 rpm for 15 to 30 minutes. For dosage forms with poorly water soluble drugs, proposed dissolution media for these kind of dosage forms are biorelevant media. These biorelevant media are suitable for carrying out dissolution studies of poorly soluble drugs and assessing the effect of food on drug absorption. The dissolution rate of poorly soluble drugs such as griseofulvin, cyclosporin, etc. is better in passive and passive than simple aqueous buffers because of the increased wetting of the drug surface and micellar solubilization of the drug. Now, the biorelevant media includes fasted state simulated gastric fluid, fat state simulated gastric fluid, fasted state simulated intestinal fluid, that is passive, and fat state simulated intestinal fluid, that is passive. Next is different approaches for enhancing drug dissolution. Now, the first and the most common approach for enhancing drug dissolution is size reduction or micronization. Smaller the particle size, the greater the effective particle size and hence higher dissolution rate. So, micronization and other particle size reduction techniques can result in higher absorption rate for drugs if dissolution rate is rate limiting step. Next is formation of salt form of drug. For weakly acidic and basic drugs, dissolution rate can be increased by changing its chemical form into basic salts and acidic salts respectively. Hence, salts form of drugs are chosen to improve bioavailability. Next is their crystal formation. Metastable polymorph usually show greater dissolution rate as compared to its other polymorph form, as illustrated by chloramphenicol palmitate, in which metastable polymorph B shows greater bioavailability than stable polymorph A and unstable polymorph C. Also, amorphous form show greater dissolution rate than crystalline form, as amorphous form dissolve more rapidly. Next is complexation. Complexation of poorly soluble drugs with complexing agents such as cyclodextrin can increase drug solubility and hence its dissolution. Next is use of co-solvents. Co-solvent are water miscible organic solvents that are used to increase the solubility of poorly water soluble drugs. Weak electrolytes and non-polar molecules have a poor water solubility. Their solubility usually can be increased by the addition of water miscible solvent in which the drug has good solubility, and this process is known as co-solvency, and the solvents used in combination to increase the solubility of solute are known as co-solvents. Commonly used co-solvents include ethanol, propylene glycol, polyethylene glycol. The solubilization of drugs in co-solvents can be used to improve the dissolution of poorly soluble drugs. Next is use of surfactants. Surfactant reduces the hydrophobicity of the drug by reducing interfacial or surface tension, and hence it increases the solubility of drug. Next approach is prodrug. Prodrug approach is utilized to improve the bioavailability of drug which undergoes extensive metabolism. Next is solid dispersion. Solid dispersion approach is one of the most commonly used approach to improve the oral bioavailability of poorly water soluble drugs. Solid dispersion is the process of dispersing one or more active ingredient in an inert matrix in the solid state, which can cause increase in dissolution and improve stability of the drug. Solid dispersion is generally used to reduce particle size. It also decreases the crystalline structure of drug into amorphous form. Next is nanosuspension. Nanosuspension approach can be used to improve the bioavailability of the poorly soluble drug. Various methods utilized for preparation of nanosuspension include precipitation, homogenization, etc. So these are some of the various approaches which are used alone or in combination to enhance the solubility of the poorly soluble drug, which in turn will improve the dissolution rate and the bioavailability. Next is in vitro in vivo evaluation method. In vitro in vivo evaluation can be done by convolution and deconvolution method. A plasma drug concentration time profile can be represented as the cumulative effect of two simultaneous processes. Absorption of the drug from the gastrointestinal tract, which is proportional to drug dissolution, and elimination of the drug from the blood. Mathematically, these curves are known as functions: absorption as input x, the plasma concentration as output y or dependent variable, and elimination as the function. The method is similar to that of linear regression used to prepare calibration curves and then using it to determine the unknown concentration either x or y depending on known variables. Similarly, plasma concentration or absorption can be known. Convolution model utilizes the drug concentration time profile obtained from dissolution or absorption to establish the plasma concentration parameters Cmax and AUC for in vivo bioavailability bioequivalence assessment. So in the equation it is given how can we mathematically write the convolution, in which Ct is plasma drug concentration after oral dose, C delta T is plasma concentration after IV dose or dose of oral solution. So these three curves in this slide are schematic representation of deconvolution and convolution processes. Now deconvolution model. Deconvolution is a numerical method used to quantify the time course of drug input using a mathematical model based on the convolution integral. On the basis of comparative data of in vivo dissolution profile, which is obtained from the plasma profile with in vitro dissolution profile, deconvolution is carried out. A deconvolution based IV-IVC model is generated, determined using a two-stage approach. First is deconvolution calculation to estimate the time course of in vivo absorption, and secondly comparison with in vitro fraction released of drug. So the IV-IVC is assessed and validated by statistically comparing the predicted with the observed plasma levels. Next is bioavailability studies for IV-IVC. A bioavailability should be performed to characterize the plasma concentration versus time profile for each of the formulation. Crossover studies or parallel studies can be carried out for IV-IVC development. It should be performed with sufficient number of subjects, that is n is equal to 6 to 30, to characterize adequately the performance of the drug product under the study. The reference product in developing an IV-IVC may be an intravenous solution, an aqueous oral solution, or an immediate release product. Bioavailability studies for IV-IVC development are usually carried out in the fasted state, as in the fed state drug absorption could be influenced by a number of in vivo variables. To develop a correlation, one of the most appropriate method is deconvolution technique such as Wagner-Nelson procedure or Lou-Rigelman method or Wagner-Nelson, which can estimate the in vivo absorption or dissolution time course. Now there are four types of IV-IVC correlation: level A, level B, level C, and level D correlation. Level A correlation. This is the highest level of correlation in which a linear point-to-point relationship can be drawn between in vitro dissolution rate and in vivo input rate of the drug from the dosage form. A purpose of level A correlation is to define a direct relationship between in vivo data such that measurement of in vitro dissolution rate alone is sufficient to determine the biopharmaceutical rate of dosage form. Next is level B correlation. A level B IV-IVC is based on principles of statistical moment analysis. The mean in vitro dissolution time MDT in vitro of the product is compared to either mean in vivo residence time or the mean in vivo dissolution time MDT vivo to quantify the correlation. Although a level B correlation uses all of the in vitro and in vivo data, it is not considered to be a point-to-point correlation, since different there will be many in vivo curves that will produce similar mean residence time values. A level B correlation does not distinctively represent the actual in vivo plasma level curves. Therefore, a level B correlation is insufficient to justify change in formulation parameters. Now next is level C correlation. Level C correlation is a weak single point relationship with partial or no representation of dissolution or plasma profile. It compares the amount of drug dissolved at one dissolution time point to one pharmacokinetic parameter such as AUC, Tmax or Cmax. Hence, it denotes a single point correlation and does not reflect the entire shape of the plasma drug concentration curve. Level C correlations can be useful in preliminary pre-formulation development when pilot formulations are being evaluated. The information may be useful in formulation development, but biowaiver is generally not possible. Now, multiple level C correlation. Due to the involvement of multiple time points multiple times, multiple level C is more reproducible than level C. It relates one or many pharmacokinetic parameters to the quantity of drug dissolved at various time points. A relationship should be demonstrated at each time point at the same parameter such that effect on the in vivo performance of any change in dissolution can be assessed. Now, level D correlation. Level D correlation is rank order informal correlation, a qualitative analysis helpful in the pre-formulation study and formulation development. However, it is not considered useful for regulatory purposes. Now last is the application of IV-IVC. IV-IVC is used to justify the therapeutic value of the product. A good correlation signifies proper release specification of the formulation. It serves as a surrogate of in vivo bioequivalence study and assists in supporting biowaivers. It validates the use of dissolution method and specification and assists in quality control during manufacturing and selecting appropriate formulations. It is used to minimize the number of bioequivalence studies performed during the initial approval process and during the scaling up and post-approval changes. Reduction of regulatory burden for additional in vivo experiment under certain conditions by performing IV-IVC as a substitute. So students, in the present module, what all we have learned is, firstly, we have learned what is solubility and what is dissolution, what is the difference between these two. Then we have learned about biopharmaceutical classification system. We have learned the different categories, different classes of drugs, class 1, 2, 3 and 4. Then we have learned about the dissolution testing objectives and its application. Then we have learned about the factors that affect the dissolution of drug. We have also learned that the different types of dissolution apparatus, their different applications, their different types and methods and different approaches for enhancing the drug dissolution. In the last, we have also learned about the IV-IVC correlation.